Harmony?
Yeah, you're good. I'm going to intro first. So tonight, as I mentioned, we have a really special occurrence where Standard Process Los Angeles and Standard Process South Coastal are coming together tonight for the September Catalyst event. We have Martha Hall with us tonight for a really special topic that is of interest to so many, and also specifically personally to me. I'm really looking forward to this. It's all about methylation and some of the genetic variances that play a role in that. So I want to briefly just kind of give you some background information about Dr. Martha Hall. She is a California board licensed acupuncturist and herbalist, and she received a master's in science and traditional Chinese medicine, a doctorate in acupuncture and oriental medicine. She is a certified clinical nutritionist and belongs to the International Foundation for Nutrition and Health. Her goal is to inspire other doctors and healthcare professionals in finding a holistic approach in healing the body with methylation, muscle testing, nutrition, and health. She's been certified in the Holistic Methylation Program, where she implements this practice into her clinic daily. So I'm super excited. Paul, you have more to share, I know, before we pass it off to her. So I'll let you have a moment to speak as well.
Pass the baton here in just a second.
Yes.
First, and thank you so much, Harmony. There's taken a significant amount of coordination and teamwork to put together tonight's presentation. I'll be fairly succinct with my overview. I did want to make a special thanks to Harmony, who's done an incredible job in sort of organizing all this with email blasts. The LA region with Steve Paniotu, David Combs, just phenomenal team. And then of course with Standard Process South Coastal with Chris Lippy, who's on, Ted Hahn, and then Melissa Bellis, who was just instrumental in so much of the PowerPoint. So let's just take a brief moment, sort of take a deep breath in. We've had close to 200 registrations, and the good news is that we are recording tonight's webinar, so you'll be able to have access it. And I was teasing Dr. Hall that you'll be able to have her visual in front of you for the rest of your life. Which is always a challenge for all of us. So, you know, tonight's an opportunity, I think, with all this going on to sort of expand and recreate your vision of what practice and patient management is. You know, Dr. Hall, and you know, today more than ever, I mean, your patients want leadership, and they not only want clinical solutions, but they want to have the development of clinical nutritional strategies, you know, encompassing clinical nutrition, on having you understand their health goals, exercise, diet, etc. And so Dr. Martha Hall is a leader. And I think when I use the word leadership, in her communication, and she'll do so tonight, she passes the baton of sovereignty to the patient so that they can take full ownership of who they are and where they're going. And so I would share with you tonight, you know, our numbers are really strong. We're united on so many different aspects. Our paradigm to understand what true health is paramount to our success, because we're all in this together. So Harmony, I'll ask you to sort of pass the baton back to Dr. Martha Hall, but I will share this. Take notes tonight, as Harmony had shared. You're going to have an opportunity to look at genetic testing, because it's here to stay. There are many different variables of it. Tonight's sort of a nice moderate to intermediate overview of genetic testing and methylation. And Dr. Hall always provides some clinical pearls that are just amazing. So relax, enjoy, create, and then we can move forward. So go ahead.
Yes, perfect. So Martha, you're more than welcome now to share your screen and get started.
Okay, so we practice this earlier.
Good.
Yeah, all good.
Share screen.
Good.
And PowerPoint. Got it? We on?
Perfect.
We are, and then just maximize that window and we're good to go. Perfect.
Nice. Okay.
Excellent.
All right.
Woohoo!
Thank you guys for that intro, and it's a lot to cover in 1 hour. I'm going to do my very best. All I can say that covering these 3 SNPs is I'm going to give you SNPs of the 3 SNPs, because when each of these SNPs can easily be done a 1-8-hour seminar just on those. There's so much information. I just want you guys to take the take-home notes of these. I hope you guys get inspired to continue on studying and learning about methylation, because it is cutting edge. It's not going anywhere, just like Paul said. So my objective is for you guys to understand the methylation terms. It's very important, because I'm not kidding you. It took me 1 year. I've been doing this about 4 years, and I didn't quite understand it until like 3 years ago. And the terms kind of threw me off. So I literally studied, and I mean studied, and researched these terms and what it means in the definitions of methylation.
Hey, yeah.
Can't take them anywhere.
Oh my goodness.
That's funny.
We're going to cover the 3 most common SNPs and understand why methylation is so very important. And hopefully, my goal is that you guys can implement this in your practice tomorrow. I love methylation. It's very personal to me. I can't talk about it too hard, but I'm going to say it real fast just because I have to detach myself from it. My father passed from Alzheimer's. And I always feel I could have done something. I don't want you guys to feel that way. It's not, once you have a DNA that's been faulted, I call them sloppy SNPs, we can do something. And I'm going to teach you guys the 3 most common SNPs today and what we can do in our practice. So let's start with the terms. Some of these are basic. The chromosomes, you know, it's a long single coiled piece of DNA. The gene, you know, you get one from one parent. Without that wouldn't make us up. It's instructions of who we are. It's like a recipe book. An allele, that I want you to know that, because when you read the raw data that you get, they're going to have that in there. And basically, it's the genes of the same type that contain different kinds of information. They could be dominant or recessive. And the next slide will go over more in depth on that one. And the epigenetics, epi means above. So say the chemical process that can influence how our genes work with our DNA. So what does that mean? It means that we can change the genetics that have been faulted, contrary to what has been told in the past. By adding certain nutrients, we can make these DNAs that have been mutated whole again. Mutation in the terms and definition for methylation is just a distinct form resulting from a change or a structure of a gene. And the variant is very important, because that's one of the key terms that we use constantly in methylation. It's a form or version of something that differs in some respect from the other, but it's of the same thing, and I'll explain that in another slide coming here shortly. These are here basically for you guys to have. This is a great example of the difference between a gene and an allele. You can say that these parrots have the same genes, but the different alleles. And the reason is they both have feathers, so you can say that they have the genes structure to make feathers, but if you look at the parrots, they're very different. That's where the difference is in allele. You see the one on the right has a beautiful vibrant colors, where the one on the left, the white albino parrot, didn't get those alleles. It's just something for you guys to know, because this is a terminology that's used constantly with methylation. You can say that an allele is just one specific way that a gene can be physically expressed. SNPs. I like to call them sloppy SNPs, because that's what happens when they become mutated. It's a single nucleotide polymorphism. I'm going to call it SNPs from now on. And what they are is they have 4 versions, and those 4 versions of adenine, cytosine, guanine, and thymine, those are the 4 SNPs that are on the bottom bases of the genes. And when those 2 are changed, it completely changes well, alters your DNA, I should say. We'll discuss a little bit more of that in the next slides. Some of these SNPs can be severe, very severe, while others can have little effect. Some can even be beneficial. There's actually an entrepreneur gene, which is pretty cool. So I would consider that a beneficial gene to have. And I actually have that one. Yeah. Some SNPs can cause instability and increase cofactor requirement function. Cofactor, you'll find out that basically just means a mineral or a vitamin. And by adding these nutrients to the diet, it can allow a gene to all of a sudden function normally. The nutritional genomics is a major goal is to expose the genetic variation. Basically, understand how the SNPs work and figure out between the diet and disease what specifically that DNA that's been faulted, it's faulty, how to make it whole again. An enzyme, as we know, it's an enzyme, it's a biological catalyst. It's almost always a protein. And basically you need to know that it speeds up the rate of a specific chemical reaction. That's the reason why we take digestive enzymes, right? So the food doesn't rot in our stomach. We take it so it'll speed up the process of digesting. Well, when our DNA and our SNPs have become mutated, we want to take on a specific enzyme, if you will, or nutrient that will speed up the reaction of that what's not been working. Cofactor, that's us. That's where we come in. Cofactor is a non-protein substance required by a protein for biological activity. Basically, it's just typical vitamins and minerals. That's basically all it is. And I want you guys to understand that because when you guys start doing the methylation, these are the terms that you'll be seeing quite often. So much to share. I want to keep going. And that was just for you guys to have and understand. What is DNA made up of? It's made up of 3 building blocks of a phosphate group, a sugar group, and one of the 4 nitrogen bases, which I just discussed. The adenine, again, I want you guys to get that in your head, the thymine, the guanine, and the cytosine. That's what it's made of. And as you see this little picture down here, I don't know if you can see my cursor or not.
Yes, we can.
Okay, good. So you see there's the cell right here. Inside the cell, we have a chromosome. Inside that chromosome, we have the DNA. That's what makes us who we are. And then we have the nucleotides. Those nucleotides are the 4 bases that will have our SNPs mutated when we don't take care of, I shouldn't say we don't take care of ourselves, it's just stress that adds it. And stress can be something that's been chemically put in our bodies. Stress can be a physical stress. Stress can be even a pregnancy. Think about it, you're going through 9 months of a complete change. That's a lot of stress on the body. So when that happens, these SNPs become mutated and we say they stops working. And on those bottoms of those bases, you'll see this number right here, ATCGTT. Well, those last 2 numbers, that's the SNPs. That's what this whole talk is about. Those tell the numbers, how to make blue eyes, brown eyes, color of hair, tall, short. That's what those SNPs are coding for us. Okay. Oh, I put this in there. The complete DNA instruction book says it contains about 3 billion bases. Those 4 nucleotides right there can create 3 billion bases. That's amazing. And about 20,000 genes on 23 pairs of chromosomes. Well, that was in 2018. Now in 2020, there's 25,000 genes in just 2 years. Can you imagine what's going to happen in 10 years from now? I'm telling you, this methylation is going nowhere. DNA, as we know, I just basically want you to know it's a sequence that codes for a specific trait or protein. And DNA and ribonucleic acid, those are nucleic acids. The way I look at it is, see that double helix right there? I was thinking about it. I took a trip this weekend to go to Texas, and my kids called and say it's cold here. So I grabbed a sweater and I noticed there's a snag on it. So I cut it with scissors as you're supposed to, because you never pull a snag because it unravels. And then I've been told you can't really fix it once it gets there. So I start to think about it. That could be just like our DNA. Our DNA, say we have a snag in our sweater, and we can't see it because it's in the back. And it's our favorite sweater, and we wear it every single day. And we wear nothing underneath it, just that sweater. And we go out, and say we live out in Buffalo, New York, where it's freezing. And all of a sudden we start to get cold. And we don't think anything of it. And we start to get weak because we're not covered, we're not protected. Chinese medicine is a big deal about going out in the cold without a sweater. Well, then we wear it often and often, and then all of a sudden we get a snag in the front. And then we'll get a snag on the side of a sweater, and finally we see it with our own eyes. And so we start to worry about it because now we start to get these holes in our DNA. Well, we have a friend that says, hey, take it to this one cleaner. So we take the sweater to the cleaner, and the cleaner, well, it cleans it. You get it back a few days later, and you look at it, and you say, it still has the holes. The cleaners say, I cleaned it. That's what I do. Well, you're upset. And then another friend tells you about a seamstress who can mend it. Well, that's us. That's us mending that DNA, those SNPs that have been snagged. It can be changed. Contrary to what Western has said in the past, it can be changed. That's where we come in. It's very, very important to know the difference between a homozygous, which means the same, and heterozygous, which means different. There's 2 copies of each gene, and we get one from each parent. Sometimes these genes are referred to as an allele. I'm going to refer to it as a gene, because that's how we recognize it as. So if a mutation occurs in just one of the genes, then that's considered a heterozygous. But on the other hand, if you happen to have copies that are from both parents and they're both mutated, then that's a homozygous type of gene. Majority of hereditary disorders are harmful if both copies are affected. I say mutated, which means protein products from both genes may falter and they're not going to work properly. So in a heterozygous genotype, one copy of the gene can produce fine proteins. Thus, these individuals are usually not affected. They're just carriers. But if you have more than one of those heterozygous genes, like you have 5 of them, well, then it can be something to look at, because when you have too many of a SNP of the same number, that'll cause problems as well. When you have a homozygous gene, it's usually 70%, it could be faltered. Here's another picture. You see these 2 colors right here. That means it's homozygous. And then below here, you see that it's hetero. This down here is for your information. You don't need to know it, but if you want to understand and read like I did, the raw DNA, you'll see that a plus and a minus sign stands for heterozygous, and I'll show you coming up, I believe, in the next slide. A homozygous gene is a plus and a plus, and if you haven't inherited a particular gene variant, then it's a negative negative. Again, that's for you. Here you see here's the gene. This right here is called a variation. Sometimes you'll see RSID, and that RSID stands for rapid statin ID. That's the numbers at the end of this gene. Oh, actually it's right here, sorry, different paper. And here's the alleles. And these colors, oops, sorry. These colors over here, yellow usually means hetero, red means homozygous. So most of them are always color coded the same, and green usually means that you don't have that variant. So when I look at these reports, which we'll teach you in a minute, the reports, I'm looking for the colors. I'm mainly looking for the red colors, which means that patient has a 70% chance of that gene being altered. And yellow just means 40% chance. Okay, that's, again, this is for you. This was done by Genetic Genie. It's a free site where you can put in your raw data, and they'll give you some of the SNPs, not all of them, but the ones that we're talking today, the VDR, the COMT, and the MTHFR gene. This right here is the main MTHFR gene that I'll be discussing today. As you see, this person right here only has a heterozygous gene. Okay. Paul, I'm going to ask you to talk about Evexia diagnostics.
Thank you. So Dr. Hall did discuss raw data, and that's sort of another subset, but you can actually, for those that have taken either Ancestry or 23andMe to look at their family heritage, which is wonderful, there is a segment that you can have access to your raw data. And I've downloaded that many times. It's almost like 10 megabyte of information. It can take a little bit of time to upload, but that's goes through so many different variables of who we are from our complete genetic makeup. It's a fascinating area that we'll have discussion for in the future. Okay, so we're really fortunate that we have a wonderful business partner. Can you go back a screen, I think?
Sorry.
Sorry. Okay, good. We have a wonderful business partner called Evexia Diagnostics. And what we've been able to do is work out some wonderful programs for genetic testing to be able to utilize standard process and, of course, MediHerb. Now, Evexia Diagnostics, many of you know Dr. David Hogsett, who does a lot of national teaching here within our organization. And the beauty of this is that he's put together several blood chemistry panels at discounted prices that will, again, utilize the standard process and MediHerb. So if you have blood chemistry analysis, you can use Evexia, and then they'll provide protocols within that. Now, from a genetic standpoint, and this is what we've been working on here for the past few months, sort of behind the scenes, there's just been so much activity in this, and Evexia really wants to be able to work with us because they also realize that that the genetics testing is going to be sort of paramount, not only for levels of prevention, but once we have been able to sort of understand that there's a genetic variance, this is a long-term health strategy for your patients. So you'll have these patients for long-term. So within this, we've been able to develop a partnership with Evexia and a sub-partner called Evexia Labs, and they're one of the premier leaders in genetic testing. So why don't we move forward here, Martha? Okay. So there's a wonderful promotion that we've been able to put together for those of you on here this evening, and Dr. Martha Hall has, she worked with Evexia as well. So if you go to Evexia Diagnostics, the normal rate to develop an account or register with them is $250, and they've taken off $150 off your registration, which really allows you to be able to move forward and have an account. As a practitioner, you just have to enter in the promotion code standard, and then you're ready to go. So you might want to write that one down, and it's evexiadiagnostics.com, and then fill out an account profile. Next, Dr. Paul. Okay. So we've been able to work through, and we're going to see a myriad of a broader or more comprehensive level of testing here in the next few months, because they're very much aware that we're doing our webinar here this evening, and they're really behind us 100%. This is just a very small fraction of the genetic testing that they are doing, and one of those is APOE, and that's sort of a sign of Alzheimer's, which Dr. Hall had discussed from a family matter here within her introduction, and maybe there'll be time to do that in another webinar. But you'll see the pricing on this, and the pricing, again, is discounted pricing from Evexia. One of those is CompT, and you think to yourself, Dr. Hall will be going through that this evening. And that's sort of the breakdown of catecholamines. And catecholamines are sort of an enzyme that breaks down our neurotransmitters and hormones. And it's really important. I have this as a genetic SNP, and if we can all be very open here this evening, I have a level of impatience. So if I have to stop or wait at a traffic light for more than a minute, you know, I sort of get a little bit, you know, I'm impatient. My wife is just, you know, calm down. So all these things sort of are persona here. And then the MTHFR is $75, and we'll be spending a fair amount of time here this evening. And again, we're seeing such a very high level of almost, it will be over 50%, Dr. Hall will go through that have this particular genetic variation or SNP. And we're seeing an advent today of so many patients that are perhaps taking prescribed or non-prescribed drugs or having levels of anxiety and depression. And when you ask them how long they've had anxiety, depression, they're going to say for a very long time period. And there you have the capacity, and we have the capability. I got this from Dr. Scott Virgil. There's a wonderful reflex point to test the MTHFR, which we'll go through at another time, but it's invaluable before we perhaps move them into genetic testing, where you can jump right into this. And then the MTHFR and COMT is $87, and it's just a great deal. And all you have to do essentially is just Evexia will send your patient or yourself those panels, and then you can, the boxes for testing, salivary testing, and then you can move forward. And then finally here, Dr. Hall will be going through his nutritional deficiencies, and this will cover the VDR, which will be fascinating overview here tonight, and how that impacts the immune system as well. Next slide, Dr. Hall. Okay, so real briefly, when you set up an account, this will be the last slide here, for genetic testing with Evexia, there's a test menu, and then you'll see Kashi Clinical Laboratories. And through that domain, you'll see, again, a broad spectrum of different testing for genetics. So it's a sort of a wonderful one-stop opportunity for you as a practitioner to be able to utilize. And again, keep in mind that they'll be expanding that as we move forward. Okay, I think that's about it, Dr. Hall. So back to you, and thank you.
Yeah. All right, let's talk about methylation and what it is. Methylation, basically, you have the terminology here, but Paul told me yesterday or a few days ago, faulty genes make faulty enzymes. That's exactly putting in what methylation is. Methylation is a body being able to detox. And when we're not methylating is when we start to have problems. We can go on to the next slide here, and you'll see things that the methylation pathway does. It's a pathway of detoxification. It balances your cholesterol. You ever have a patient that has high cholesterol? Not me, but I have a patient that has high cholesterol. And he came to me and says, I was referred to you. He said some name I didn't know. And he says that his cholesterol, I said, I could help you. And he says, yeah, my cholesterol is in the 500s. I said, oh, no. I said, I don't want you having a stroke on my watch. I'm sorry, I can't take you. You need to be on the statin. Now it's different. He says, actually, so-and-so at the hospital near me referred him to me because he's severely allergic to all statins. And so I said, okay, well, in that case, I can help you. And it's actually been really good positive results with him. His cholesterol's in the 300 and he's doing fine. He obviously has this methylation issue, but remember, cholesterol in the 70s were in the 300s and we were fine. We didn't have the heart disease that we have now. The main thing about methylation is to detox heavy metals. And we'll discuss about that. I like this one because I know you guys just want to know what it is, what it does, and how you guys can help your patients. This is the simple version, right? If DNA is turned on, then you guys are methylating, the body's working properly. If it's turned off, nothing's happening. Well, I shouldn't say nothing happening. You start to get a cascade of things starting to falter, not work properly. So they discovered the MTHFR gene back in the 2003 when the Nutritional Health Institute got together and they wanted to know how many DNA, how many genes does the human body have? I mean, what a great project, right? I like to be a little fly on the wall in that meeting because they came up with so many gene variants. And one of the two variants that they came up was the MTHFR. So again, think about that. This hasn't been around that long. Oh, another note, I want you guys to know that all the information that I received here has been backed up. I did look it up. I did look up on PubMed and SNPpedia, which is part of the PubMed. And it was all backed by what I'm saying on here. There's a few things I was looking for. I didn't quite find it. I didn't put it in here, but those will be pearls that I'll tell you about. This is something that I didn't know. 45% of the population has a copy of it, and especially Mexicans, Italians, and Chinese. I have the heterozygous genes, and both my kids have that as well as my mom, but we don't have the homozygous gene, which means it's more of a 70% chance of that gene being mutated. And these are just some of the things, just some of them, of what this gene is in charge of. Multiple miscarriages. I discussed this in 3 slides, and I really want you guys to get that in your head. The reason is because I've had patients that had multiple miscarriages and they never got better. And I feel bad. I had to send them somewhere else. Fertility is not my specialty. And I figured maybe because that's why I didn't know, but now I understand what's going on. They went to fertility, they went to other doctors, and they didn't get the help, no answers, I should say. Alzheimer's disease, as we discussed, dementia, mood disorders, PAD, peripheral artery disease. These are the patients that are coming into our clinic, right? Acute leukemia, that's one that I have seen, and I never understood that one either. My niece's very best friend got the vaccine, the human papillomavirus, HPV vaccine. Her mom took her when she was, I think, 14. And within 6 months, she got acute leukemia. And I remember seeing this little girl grow up, and I was so devastated. By then I moved. I used to live next to my brother, and then I moved. And when I found that out, it was just devastating. But most people that I know last a long time with leukemia, where she only lasted 2 years. She died within 2 years. The sad part is 2 months ago, my niece called me up because her mother, the girl that died, asked her to call me and ask if it was her fault that her daughter died. There's no way. I told her, don't do that to the mom, let it go, it's too late. Well, her mother found out about the MTHFR gene, found out she was a carrier, and then she started putting the pieces together. And she called like 3 times, and I said, we don't know, who knows what happened, but she was perfectly fine before that. So that's acute leukemia. I'm sure you guys seen it. I'm sure you guys heard about somebody getting acute leukemia. I've seen it in my practice 4 times now. And this is before I understood the methylation. Nobody should just get acute leukemia. Okay, here is 2 of the SNPs or variants. Remember, it's the same thing, but it's the same MTHFR gene, but these numbers over here make it different, a variant. When you have the heterozygous gene of this SNP over here, which ends in 7T, you have a 40% chance loss of function. When you have a homozygous gene, you have a 75% loss of function when it gets mutated. Just because you have that gene, it doesn't mean that it's been mutated. Something has to cause it, some stress has to cause it to mutate. But this one, I can tell you, the 7T homozygous gene, that's a very, very important one to look to see if your patients have that. If they have that, it's very important that they do not get vaccinated when they are sick. If they can avoid it altogether, it's extremely important because one weakness in their immune system was going to bring that 75% loss of function up to a higher level now. If you have the other SNP, that's not so much with birth defects as the 7T one is. So as you see this SNP over here, 8C, you only have a 20% loss of function. Where we get the homozygous, a gene from both parents, it's a 40% loss of function. So it's a big difference. This is important to see over here. When you have compound genes from both parents, when you have compound heterozygous genes, which means the yellow lines, you only get from one parent. But if you got many of those variants, you're still going to have a 40% chance loss of function. So that doubles that amount as opposed to 20 to 40. And here's another little pearl. The 7T variant will increase homocysteine levels. How many of you guys have patients that have high homocysteine levels and yet their diet is good? I have plenty of them, and I never understood this. Now I do. So now when I see it, I'm like, yes, I know what to do with these patients. And the 98C will not have an effect on it. Okay, so this is the one that the 7T gene. I want you to know that when the patients start having multiple miscarriage again, it can be a mutated MTHFR gene. And what happens when it starts to it becomes undermethylated, meaning it's not doing its job. So you start to become accumulation of toxins, heavy metals, chemicals. You start to get sick more often, and you're just not getting well. Allergies, and when I mean allergies, they never had it before. And all of a sudden, everything, they're sensitive to everything. This is, start looking into this and given the products that we'll discuss shortly. So the 6,7-T gene converts homocysteine to methionine. And when it's mutated, basically you can see the high levels of homocysteine. Okay, here's a pearl. This is really important for you guys. This is going to show up on their blood labs. So when you see this is what I've always seen in the past, and I couldn't figure them out. It would drive me nuts. I know this one case in particular, the guy did yoga. He worked out 5 days a week. He ran. And he lifted weights, so he had a great balance of everything, but why high levels of homocysteine? Well, now I know why. So we had his DNA ran, and then we figured out he had this gene. He's doing fine, but I still have him checked every 9 months with the blood work. I believe in having labs because it's another piece of the puzzle. Dr. Ben Lynch, I actually love that book. I have it on my phone. I have it on my iPad, my Kindle, and my audiobook. And this is, can you see? Oh, I don't know if you can see, maybe not.
Not good.
That's interesting. Okay, you can for whatever reason it's not showing, but it's Dirty Genes. It's a good book, but he talks about what foods you can eat. The problem is when you go to the store and you try buying the foods like leafy green vegetables, well, they're not pure like they used to be 25 years ago. When you buy a tomato, it doesn't have not even a quarter of the nutrients that it had 25 years ago because our soil is not the same. So anyways, Dr. Ben Lynch, who's one of the founders of putting SNPs out there, says that when you get an apparent gene of the 6,7-T, you'll have symptoms like autism. Again, we'll discuss that a little later. Chronic fatigue syndrome, fibromyalgia, miscarriages, again, IBS, you're gonna have birth defects, you're gonna have Alzheimer's, bipolar. I can keep reading. This is just what he says, and there's so many other conditions. Are you guys getting me here? Do you understand that these are our patients that are coming to see us? Do you understand that some patients can't even be touched? And the sad part is they're coming, they're paying you money to help them because somebody else says that it was in their mind. It's not in their mind. Why would anybody pay you money for you to tell them, oh, it's just, you're fine? They want to get help. I didn't understand it now. So people that are hurting and can't even to the touch, they hurt. They're very, very sensitive. This gene has been severely mutated by then. This is the other variant of the MTHFR, the 98C. This helps generate BH4, which is found in the cataplex speed core. You probably can't see that either. It's a new product, and we'll discuss that in a minute. But BH4 is really important for detoxing of ammonia, production of neurotransmitters, brain issues, those people that you see walking the streets talking to themselves. Sad, but I guarantee they probably have some kind of genetic gene that's been mutated. BH4 is also helps with the production of nitric oxide, which we know has to do with cardiovascular. Very, very important. So when somebody's having problems, always go back to the source. Start asking questions. Where does this problem start? And that's what I do. I actually kind of enjoy it. I love logic problems since I was a kid, those puzzle games. Well, this to me is another one of those puzzles. Main thing with this gene is mental emotional symptoms. Mercury, lead, and aluminum will act as a drain on this. Where do we see mercury, lead, and aluminum? I don't know, maybe the vaccines that are out there? Just saying. Here's a pearl. Far infrared sauna promotes detoxification, and that helps increase the BH4 levels. I didn't know that. I thought it was a little fun fact there. Don't get discouraged by this. I tried to find the simplest map, and this was one of the simplest one. And the way I ask you to read this or understand this is look at each little, this is called a methylation cycle right here. Look at each one. Don't look at the whole picture because it can get confusing. So we are talking today about this SNP right here, VDR, this SNP right here, MTHFR, and this SNP over here, COMP. You see how many other SNPs there are? There's a lot. These three right here are my favorite. In order, VDR. I would probably say COMP and then MTHFR. I know those last two are up for debate. But the way it works is when you're methylating, the cycle goes like this. And then all of a sudden you get lead and it gets thrown in there and it comes to a halt. It's not functioning anymore. And let's say that your vitamin D levels are low because you have that VDR gene that we'll discuss in a bit. Well, now it really starts to cascade down the problems. As you see here, the VDR is linked to dopamine, which also has to do with the feel good hormones, the people that have problems walking, talking to themselves down the street. And you see the people that are about to lose it also. I see this issue with menopausal women. They're not crazy, but they feel they're going crazy, and they literally tell me that. They're losing their DNA gets faulty because of the stress of what's happening during menopause. Two of my brothers got divorced during when their wives were going through menopause, and now I understand. You just get uncomfortable and everything bothers you. And now I'm seeing it more with women than with men. They used to say men going through midlife crisis. I say it's the women now going more through the midlife crisis. Anyways, this is just here for you. I wanted you to see, do not read the whole thing. Don't get discouraged. Look at one section at a time. That's it. Just look at one section at a time. And another thing, you're going to see different variations. There's that word again. Different variants of these map. So since it's cutting edge, don't just think this map is the correct one, because there's other ones that are up for debate as well. Beautiful. Chelico is a product that I test. There's not a day goes by in my clinic that I don't muscle test, because I muscle test my patients to see if Chelico is a product that they need. Chelico is a beautiful product in detoxing the patients that have the heavy metals. It has hawthorn, which is beautiful for your cardiovascular patients that have health issues. Methylation, as we know, is great for the liver. My patients, I have quite a few that have cirrhosis of the liver, and a lot have fatty liver. Methylation is my go-to. And garlic, as we know, is a beautiful product for antiviral. But Chelico, I give this product to my patients to pull the heavy metals, the chemicals out of the body, especially, actually, they've been vaccinated. Oh, okay. Next one is choline. Choline, as you'll read in some books, is known as B4, vitamin B4. And it's also in the new product called Cataplex B Core as well. This choline is one of the most important products. I want you to let that sink in. Choline is one of the most important methylation supplements that we have. It's extremely important for opening up. Remember that map that I showed you? It's extremely important when you have that kink, when the methylation is going like this and you get a kink in there and it stops functioning, choline helps open up those pathways. It's something that you should start your patients off with slowly, maybe 1 twice a day, and then you move up until I'll be doing two tablets 8 times a day after I make sure they're okay and make sure that their body starts functioning like it should be. And I have them check in with me daily just to make sure they're doing okay. Cataplex B Core, yay. My favorite product is before this was Cataplex B from Standard Process. Used to be Zypen, then it got bumped down to Cataplex B. Cataplex B for me, and remember, helps to do with brain, helps me chill. It helps me relax. When I just get a little anxious, yes, I do have that anxiousness sometimes. I didn't think I did, but when I started taking Cataplex B, that went away it goes away within minutes. Now Core, because it's fewer products and there are fewer supplements, you don't need it. I think 2 at once is a lot. Spread it out. I would do 1 twice a day. It's important because it has folate in there. And what's the problem with the MTHFR? The folate is involved in that. They get low and they can't absorb the folate. They can't break down the folate. So this is a whole food source where the patient can take. Thiamine is very important, right? We talked about thiamine or the 4 bases. It is part of the 4 bases of the nucleotides that makes up those SNPs. This helps keep those SNPs functioning like they should, so you don't get a snag or sloppy SNPs, as I say. Okay, make sure we're good on time. The comp gene, the comp gene. This is so important because I always say, oh, my comp gene is acting up, but only to the people that understand methylation. These are the ones that get uptight, and like Paul said, he gets a little irritable at the stop sign or a signal light. Takes too long, right? Yeah. Those people that can't even wait 3 or 5 minutes, and that's when you know the comp gene is starting to act up. But here's the thing, when the patients that you see or children that you see that have ADD or ADHD, usually there's an imbalance between the epinephrine and the dopamine receptors. So because COMT is in charge of breaking those 2 down, and when it's not breaking down because they're not methylating, the COMT gene starts to become mutated. It's involved with personality. People say we joke around saying it's a COMT person. His COMT is acting up. And with the mutated homozygous gene, that means for both your parents, you're going to see irritability, hyperactivity, just basically abnormal behavior. Parkinson's, this usually shows up later. It won't show up with the children. It shows up later with your patients. They were perfectly fine. There's no Parkinson's involved in their family in the past, and all of a sudden they have it. Start looking into this. Let's start getting them methylated and adding the products like choline, like the products that we'll discuss soon as well. Imbalance of estrogen levels. Yes, you see that quite often. Remember I talked about a menopause woman? Yes, that's where the balance gets. And low pain levels, those patients that can't even handle being touched. This is one of the things that the comp gene you want to look at. Methylation shake, I love that. Why? Because it has all the 9 essential amino acids. You can read everything that has in here, but it's something that I believe all families should have a shake in their house. Why? Because there's that day where you just don't have time to have a good, solid breakfast, a healthy breakfast. Where else are you going to get all these products in the shake? I've been doing shakes before I was, I think I started when I was playing soccer in college. I've always done shakes. So this shake is absolutely beautiful, and it's vegetarian. So if for your vegan and vegetarian patients, this is a great product to add. Easy Mag. Easy Mag, as we know, magnesium is so important in the body. It's a precursor to 300 enzymes. So without magnesium in the body, we're going to start to have a lot of enzymes. Remember, we talked about the enzymes. It's a catalyst that makes things moving. It's not going to go. Magnesium, if it's low, you're going to start to have issues. And the main ones that you'll see usually are cramps. That's one. Even the hair loss you'll see with the magnesium. So I like this one because it's easy. If kids that can't swallow, or even adults, yes, I have adults that can't swallow. I have a friend of mine who won't take patients that won't take supplements, that won't swallow. He actually just sends them to me and hopes that I can make them a formula or something. But Easy Mag is a great source. Also, more importantly, for right here, K1. We're going to discuss the VDR gene. You want to have K with the VDR gene, and we'll discuss that shortly. Here is for you guys to have. It's talking about VDR, how we get the vitamin D. In a perfect world, we go out in the sun with no lotion, no sunscreen on. And we, after 20 minutes of our pores being opened up, the sun comes down, opens up our pores, it gets in, it creates an enzyme, and then we get synthesize this chemical, and it goes to our liver. And then from the liver, it goes down to, it goes down to the kidney. And that's the active form of vitamin D. So you can see we don't live in a perfect world because we put sunscreen on, you know, with good rights because we're afraid of what could happen, skin cancer. And we put lotion on so our skin doesn't get dry and it looks nice and pretty. So it's hard to get this. And the older we get, we don't synthesize it quite easily. So VDR stands for vitamin D receptor, and that's the gene number right there. And it is not just important for osteoporosis and teeth and bones, it helps to do with immune. Patients that have this receptor, this variant, they can't break down vitamin D. It stays in your system, and they may take vitamin D and they're still low. So this VDR protein binds to the active form of vitamin D. And remember I said for it to be active, it has to go all the way to the kidneys. Well, how many patients of yours have digestive enzymes? How many, have digestive problems, right? So if they're having digestive problems, they're not going to get the D like it needs to be. And if they get it, they're going to be even at a low normal. Again, here is from PubMed. And every SNP, as you saw, there's 25,000, okay? There's no way I'm going to memorize these SNPs. So I have to do something in my head to memorize what each SNP is for. And the SNP that VDR, chronic disease. Any chronic issue, think vitamin D. If there was a test at the end of this Zoom meeting, I would ask you guys, what variant is responsible for chronic disease? And the number one is VDR. That's why vitamin D, the SNP, isn't the most important to me. This is a pearl for you guys to know when VDR SNP has been mutated. You don't have to have blood work is going to show, that's a given. But when the patient comes in and they say they're taking vitamin D and they're still low, bingo, you know they got that gene. They definitely have the gene if the doctor prescribed vitamin D for them, because it's still low. They're going to think, okay, I'm going to give you 30,000 milligrams of vitamin D and we're going to get that your vitamin D levels up. And they'll still be down at a low normal. They have this gene. They need help. They need the vitamin K and vitamin A with this. These are, sorry guys, these are things that happen with the vitamin D when it's been mutated. Colds constantly. You know somebody that keeps getting colds, right? And they say, oh, because it's wintertime. It's what does it call it? Flu season now. Well, why? Because there's not as much sun. And what else is going on? Halloween, so more candy. Thanksgiving, more dessert. Christmas, everything. Dessert, candy, all of the above. So sugar depletes us. We're getting no vitamin D. And if we're not taking vitamin D, our immune system goes even further down. Eczema, yes, eczema has skin disorders have to do with VDR. I have a patient that has six homozygous genes of VDR. And he has he doesn't have his top layer of his skin. And we're getting help. So I need his levels up in the hundreds. You'll see that 12 to 30 is low, which is good because before they wanted 25. You can't function at 25. But this is Western, they consider it low. Functional is what you all should be doing, myself included. We want the levels from 60 to 80 is functional. High is anything above 125. Well, guess what? There's some patients that need to be at 100. If they have chronic issues and they're not getting better, just try it. It's not going to hurt them for a short time. Get those levels up. Okay, this is another variant, the FOK. I don't know who names these things. This is related to blood sugar. Any of you guys know anybody that's got type 2 diabetes all of a sudden and for no reason? You know their diet's pretty good and their diet's probably better than yours. Why all of a sudden are they getting pre-diabetic? They have that gene that's been turned on. You need to give them pancreas support like Genema, metabolic complex, Diaplex, and of course, the VDR support program that I'm going to talk to you guys about. Supplements, Cataplex D. This is a must. This is the protocol that I want you guys to take home today. It's not hard to figure out. Those VDR patients that you have, it's not hard to figure out. I'm going to tell you again, VDR patients are not hard to figure out. They have chronic issues. They're constantly getting sick. They're fatigued. They don't know why. They're a little depressed, not severe, well, maybe severely, but most of them that come to see me, not quite severely yet. So I give them a vitamin D, and I'm not kidding you. I get a text the very next day. That means they were so deprived. Vitamin D is not just for bones and teeth. Don't forget that. It's for the immune system. This vitamin D contains 300 micrograms of D, which is very important. That remember I said the vitamin D protocol needs A and K? Well, cod liver oil has the A and D in there, but it's twice the amount. So those patients that want to take, it's your choice. I kind of like the cod liver oil because it has higher amounts of vitamin A in it, or you can have them take more of the D. I'll literally, like I told you, have the patient take 2 vitamin D 3 to 4 times a day, sometimes 6, depending on how bad they are. And then I will add maybe sometimes some more vitamin A. The A is just a cofactor, okay? The A and the K is a cofactor. Cruciferous Complete here is a cofactor for the vitamin D to be activated. It's the great thing about this, there's 90 capsules, all they need is 1 a day. That's sufficient enough for them to be able to absorb their vitamin D. Okay, so remember, vitamin D, A, and K. The other 2 products have A in it, or you can do Cataplex A. I also give Cataplex A, and I have them do 1 A of that a day, along with 1 to 2 Cruciferous Complete. It depends how deficient they are. I love this. The fault is not in our stars, but in ourselves. Okay, so the fault is not in our genes, but in ourselves, right? We can change it. We can change epigenetics. We can change this by adding the proper cofactor that the body needs. Find out which gene is faulty and find out what that nutrient is needing, what SNP is needed for that nutrient. I want you guys to remember, treat the patient, not the SNP. Don't get so caught up in the SNPs that, oh, this guy has 6 SNPs of this MTHFR. Oh, my Lord, he's a mess. He may not be a mess. It may not be mutated. Just warn him casually. Don't make it seem like it's a big deal, but just warn him. And let them know you have this if ever this starts to happen. Unfortunate for me, they come in, they already have it. And my guess with you guys, it's the same thing. My cases aren't easy. I get so excited when somebody just comes in for a cold or a flu. Doesn't happen. They're coming in with severe issues, very severe issues. And I have a lot of professional athletes that have brain damage from too many blows to the head. So that comp gene has been turned on, right? And so I have them methylate. I'm working with 2 at this present time. Understand the case before looking at the SNP. So again, ask questions. Doing SNPs and reading raw DNA, it's just here to help you what nutritional deficiencies they need. It helps to recognize what may lie in their future, and also helps you create a long-term plan, and helps explain why a patient isn't responding, right? Oh, here's a thing. I do muscle testing. I say this is vitamin D. Oh, you can't see. Say that I have vitamin D in my hand, and I'm muscle testing. And I would put the vitamin D in the patient's hand. I muscle test them, and they test weak for it. I said, oh, okay, you don't need vitamin D. I used to think I didn't need vitamin D, right? Because I'm young. Well, when I was younger, not now, fortunately. So I wouldn't give the patient vitamin D, but then I get their blood work, and they say, oh, you're low on D. So what happened? Maybe it wasn't standard process that they needed. That's not the case. You give them vitamin D for your muscle testers. You put Cruciferous Complete in there. Watch what happens. They test strong as an ox. Another favorite thing for me to do is I keep the patient's legs together. And I say, don't let me spread them apart. I pull with my hands, and even the professional athletes, they get weak. When I put a proper protocol on their body, and I'm talking an 11-year-old as well, those legs won't separate. those legs are staying together, because strength is not just about physical strength. It's about core strength. It's about being healthy as well. These are references for you guys. 23andMe is a genetic testing site. I prefer Ancestry.com, which is in the next slide. Why? Because you get more bang for your buck. Because Ancestry will test for more than the 23andMe will. Snippedia.com, it's a beautiful site. All you have to do is put the number of that SNP, like the C667T, you put that into that Snippedia site, which is hooked up with PubMed, and all the variants of that show up, and the disease and the studies, if any, is shown on there. Genetic Genie is a free transcription of your raw data. Of course, we know PubMed, Promethease, and the books that I talked about. It's in order that I like. There's not that many books out there on epigenetics and methylation. Trust me, I've looked. I probably have about 8 of them. And these 4 right here are the best out there. I like the second one. It's so simple and so easy, and I really like the way she laid it out, but it's still more in depth than that. Here's the steps for Ancestry.com for you guys who, the way you do is Ancestry.com is the way this works is you order the kit from Ancestry.com, or you can go to Target and Walmart. Yep, they sell them now. I was shocked to see that they sell them. You bring the kit home. You have to create an account on your phone or your computer, and you put the number of whatever that box is on there, and now it's linked to you, and you send it in. They're going to send you, because of COVID, it's been taking over 2 months now, where you're from. You know, I was 52, I forgot, 56% from Spain, my family was. I don't care about that. This is the steps to download your raw data. With this raw data and your patient's raw data, you're going to save it onto your desktop. And from there, you can put it into other sites. Or we're working with Evexia. With Evexia, you don't have to do the Ancestry. You can take their test kits and do the saliva test at home and then get the results from there. We're still in the process of finding out who we're going to use for the genetic testing. It's not hard. I know there's people out there that don't like to get out their information, but this is mine. I'm not going to read it to you guys, but I think the negative comments that I've heard about doing the Ancestry, the positive results outweigh by far the negative comments out there. That's my opinion. I want to thank you guys. One thing I forgot to mention is that it's going to take minimum of 3 to 4 months before we start seeing results with DNA mutation. I will see results like, oh, they're not depressed anymore, but it was mild depression. It wasn't severe. So when you have something like alopecia or the hair falling out, you have Epstein-Barr Lyme's where they're having relapse after relapse. Of course, we can't make that go away because that is a stealth pathogen that lays dormant, but we can make sure it doesn't come back again. And which one we use for that one? Does anybody remember? VDR. That's a VDR protocol. Vitamin D levels. We want to keep them high. And just because the labs say up to, what did they say, the normal for labs, they would say even 40. They got to be higher than 40, people. Come on, doctors. This has to be up in the 80s, 90s. Some have to be in the hundreds for them to start functioning normal, to make sure that they're methylating properly. You guys, I want to thank you for your time. My goal is to hope that you guys get the inspiration and the drive to continue with this, because we need more doctors to understand this. This is cutting edge. You guys out there are doing amazing things. Let's keep doing that. Let's help our patients. Again, we're not doing any damage by giving them the whole food supplements. The Standard Process is my favorite, hands down, my favorite supplement company, and mainly my only company that I use to help my patients succeed in their health. Thank you. If you guys have questions, feel free to email me or to contact your rep. I promise you I won't ignore you. If I don't respond, send me another email saying, hey, you forgot or you didn't respond to me. I get about 1,500 emails every 2.5 days, so I try to go through them carefully. Thank you again, And good night.
Thank you. Thank you, Dr. Martha. And I have a few questions that have come through. I also noticed that some of you emailed me in some questions that were more of kind of case studies. So those can always be something that I forward over to you as well. But we have a few little things that I'll bring up. And if you have more questions, please send them in. But we had a question come in. What do you give a patient with the high homocysteine levels with the variant on the C677T?
You, first of all, the shake will help. The homocysteine levels, you're going to have to give some cardiac support as well. Cardio Plus is one of the products that I use, of course, because it has 4 of the top ingredients for the heart. I also give them part of the methylation. I always check every single one of my patients' VDR, every single one of them. You know how easy it is to check? Like I told you, put the vitamin D in there. If they test weak, more than likely they have that VDR SNP. If you don't muscle test, ask them, have your vitamin D levels ever been low? Send them out for blood work. If you don't send out for blood work, have them go to their doctor to get blood work. Look at it, make sure they test the vitamin D. Not all doctors check for vitamin D. So put the vitamin D in there. You do cardiac support like Cardio Plus, and along with methylation, you want choline in there to make sure. And Cataplex P is my favorite. I have I always check my patient's heart when they have high homocysteine levels because I have the machine that checks in real time. I can't think of the machine name. Paul, what's that machine? Do you know what it is?
The acoustic
Heart sound recorder.
Yeah. Heart sound.
Yeah.
I literally give them, I have them chew the Cataplex B, bam, within minutes it changes. Minutes, and I have all that.
That machine is amazing.
So Cataplex B core
That's awesome.
that as well.
Okay, perfect. We also had a question come in regarding the levels of vitamin D. So someone referenced Dr. Rhonda Nelson saying that between 60 and 80 is too high because of hypercalcemia or pulling calcium from the bones. What's your feedback on that?
Yes, that's what I said. Only some of patients will need high vitamin D levels. Those are the ones that have the mutated VDR gene. And that's why I say work with them closely and watch them pretty much every month. Vitamin D is so easy to raise as it is so easy to get lower. So all you have to do is stop. There's exactly right about the hypercalcemia, vitamin D-osis, I call it, hypervitamin-D-osis. So that's why you work with them closely. I have a patient who doesn't have skin. His top layer is sheds every few hours. He's got so many SNPs. He needs to be high vitamin D. There are exceptions. And by the way, I learned about giving patients high D way back, probably about 15 years ago. I had a doctor that taught me, she says, you will get some patients, not all patients, that will need more than the norm. We're not all the same. Right. All us functional medicine doctors out there, we know how to look at each case individually. I would not give high vitamin D to somebody that doesn't have a vitamin D receptor. You give it only to the patients that have it and they're not functioning properly.
Right. That's great. You know, it's a very individual thing, as we know, with most of this.
Yeah, and by the way, I love Rhonda Nelson. She's fabulous.
Yes. We have another one coming in that's a little more of a case basis here. She says, my business partner has tried everything, supplements, to try to methylate her body. She can't handle hardly anything for supplement support because she will start detoxing and feeling horrible. Any suggestions?
Absolutely. So I have a buddy, he's one of the top accounts of Standard Process. He said I could give him a shout out today, Dr. Rob in Newport. He always gives patients 21-day purification. That's the goal, but he does it at the end when they're healthy. Years ago, I said you should give them at the very beginning, and that's what I used to do. I literally had about 4 patients, not exaggerating, actually about 5, that drove themselves to the hospital. And I told them, do not think you're getting sick. Do not think, this is before methylation, do not think you're getting sick. You're just going to hit that wall. Well, they're not methylating. They have the MTHFR gene and they can't methylate. So you have to start them off very slow. Choline. You start with choline, and if they're that severe, that gene is being mutated, you want to start with 1 choline, twice a day and then work their way up. It's going to take them a long time to detox, and it's not going to be probably within 9 months. Have her email me, and I'd be more than happy to go over the case study with her, because those are cases that I used to see.
Okay, perfect. Well, thank you so much. I will forward over that case study with the other few that came through. As Dr. Martha Hall said, if you have additional questions, please send them in to anyone on either the SPLA team or the SP South Coastal team. We will get those answers to you or forward them to Martha for her advisement on that. And as always, the recording and the notes and any additional resources that were shared tonight will be sent out for SPLA in the Monday weekly beat. I believe they will also be going out on Monday for SP South Coastal. So you'll have your recording, your notes, and anything else that was shared tonight. I thank you all so much for joining us for this event. And again, thank you, Dr. Martha Hall. Thank you, Paul. We appreciate all the work that went into tonight. And everyone have a great evening. Thanks
Harmony,
so much.
thank you. You did all the work.
Thank you. Thank you. No.
Thank you, everyone.
Team effort here.
All right.
Have a great night.
Good night, you guys.
Bye.
Thank you.
Good night. Thank you.